All right, I think we're going to go ahead and get started. I want to welcome everyone to our new ADA Pathways seminar series. We are including both all of the current pathway awardees as well as alumni who have gone through the program and hopefully benefited a lot from the funding they received from the ADA. And the purpose is to update on the work that our awardees have done, but by including the mentor advisory group, also to engage in hopefully some good question and answer and some generation of ideas that will fuel future research in our awardee laboratories and maybe via collaborations. I just want to say that as a member of the mentor advisory group from the very beginning of this program in 2013, it's one of the most rewarding academic chores, if you will, that I've ever done, because A, the quality of the applications was sensational, and B, I think I would speak for the other members of the mentor advisory group that we really felt like we were doing something useful and impactful, because the awards that we were able to provide were substantive and really seem, in many cases that I know about, to have contributed to launch of careers of young faculty or launch of transitions mid-career faculty or people from other fields. So I just think it's been a really worthwhile endeavor. And although we're in a period now where the program is not able to be funded, at least for a limited period of time, I think it's important to keep the group together, keep the band together, and look at some of the things that have been accomplished with the hopes that this will encourage the ADA to fund this program again in the future. So the format of our lectures is going to be 2 speakers for each session. We have 2 speakers today, 20 minutes for presentation and about 10 minutes for discussion. The key to success is the discussion period. I hope it'll be vigorous and that we'll have participation both from our mentor advisory group and our awardees in asking questions and in going back and forth. So step up, please. I'd like to ask everyone to ask your questions in chat, and then I will call on you. When you're called on, Kylie Goldstein, our ADA organizer, will take you off of mute so that if you have a specific brief comment or follow-up, you'll be able to ask that live following your question. Although I will have to be on top of that a little bit in terms of timing because we want to stay within the hour. I know everybody has busy schedules. I do want to thank Kylie Goldstein at ADA for organizing this series that we're going to have, and for the very difficult task of taking over for Magda Galindo, who's been the sort of face of the Pathways program since its inception. But I want to make everyone feel good that in working with Kylie for 2 months, she's terrific, and we are in good hands in going forward. Kylie has generated a schedule for our future sessions in 2021, and we're not sure if those were attached to the calendar invite. If not, they will be coming out as an email. That schedule will be coming out as an email, so we all can keep up with the first Wednesday of every month in 2021. We're going to have a 2-person presentation event, and there is a complete schedule of that now, and either it'll be on the calendar invite or Kylie will get that to you in very short order. And in the future, we'll send reminders of the talks and with titles about a week before. And that's a prompt actually for those who will be speaking to please send your titles, your talk titles to us, send them to Kylie as soon as you feel ready to do so. All right, so that's it for the introduction, and I want to jump right in, given our limited time. We have 2 speakers today, Sumita Pennathur from UC Santa Barbara and Marie-France Hivert from Harvard. And Sumita has volunteered to kick us off, and her presentation is entitled Next Generation Technologies for the Management of Diabetes. Sumita, please.
Sumita, you're muted. Thank you. Okay, step one is done. Thanks so much. I am happy to be the first one. I'm honored to be the first one. This is my end of my fourth year as the Visionary Awardee. Just for people who don't know me, let's see if I can click through this. I've been a professor of mechanical engineering for 13 years. I started in 2007, and my main specialty is electrokinetics, driving fluids around with electric fields, actually going down to the nanoscale and really understanding what's happening at the nanoscale with electric fields and fluid moving around. I do a lot of device making. I make MEMS and microfabrication MEMS devices. I learn about how to build things better, smarter, cheaper. And I've actually been involved with biosensors. In fact, one of my companies, so on top of research, I start companies. And for example, when I was pregnant with my son, who's now 14, I built a breast pump for Medela. I designed and sold a microfluidic device to detect fetal maternal hemorrhaging, microfluidics. And the latest one was biosensing for nanofluidics, where we have Series A. In fact, I just submitted for an EUA for COVID detection, handheld COVID detection that goes to your phone. So you guys can all look at alveotechnologies.com. Hopefully they'll be out in the market soon. And most recently, during this ADA award, I started a company, Lakshmi Therapeutic Devices, for my daughter for diabetes. And I already spilled the beans here. What ended up happening is in 2014, my daughter here got diagnosed with diabetes. And since we're all working from home, if you want to look at the screen, I'm able to actually bring the guest of honor here. You want to wave to everybody? See, she's grown 6 years. And what happened was she was diagnosed 6 years ago, and you just saw my past. I started companies, and I was able to use all my research work for mechanical engineering good. So when she got diagnosed, there was no question in my mind I was going to save her life. And I have pivoted to spend all my time and effort on building technologies to make her better. So I've wanted to untether her from diabetes. You know, you can see all the tethering she has. I don't know if you can see that. And you know, obviously she's using the greatest technologies, the CGM and the Medtronic pumps and the Dexcom and whatnot. But given my past, I wanted to do something to help her. And so to be honest, this ADA Visionary Award is the only thing that really is the thing that helped me achieve this goal. So I can't thank ADA enough. This visionary program it is exactly what it wanted to do, which is get somebody that has 13 years experience doing something and then all of a sudden is passionate about diabetes for a very important reason, to use everything I have to save her life and do something better. So I wanted to do both. The visionary award was to build a better CGM. You can see the Dexcoms here and the Abbotts. They're subcutaneous. They're pretty long. Luckily, she doesn't scream as much now with the Dexcom. She still does. It's only about 5 minutes of screaming as opposed to an hour, right? And the the pumps are still hurting, right, Sophia? So I wanted to just do one. I want to do it both integrated. My vision was to create something, as you see here on the top, people are trying to make integrated devices. I wanted to make innovations to make this better. I knew I could do that. And so the first 3 years, I'm in year 4 now. The first 3 years, I spent a lot of time working on sensing the pump, the sensing. And in fact, I now have a company for sensing. Um, and so the last 2 years, I've now been focusing on the pump. So I'm going to go through what I've done the last 3 years, and I'll show you the very, very latest data that I got over the weekend for the pumping. So as I said, for sensing, and actually for pumping, I want something smaller than a penny. And you guys know the device is just the infusion set's bigger than a penny already. And you can see that I want it to be a Hello Kitty sticker. You can see how small that is. Hello Kitty sticker, that's that size compared to her Dexcom. And how do I do that? Well, I want it to be painless, disposable, affordable, small form factor, a daily application, and of course, no finger pricks. Now, 3 years ago, there were still finger pricks everywhere. Now, most CGMs don't need finger pricks, so that's less of a need. And here's the general concept. This is what we came up with at UCSB. You know, full disclosure, the company I started, Lakshmi Therapeutic Devices, was in direct competition with this, doing a different concept, completely different, but I wanted to just get as many technologies out there as possible. I figured, you know, if I'm competing against myself, I'll always win. So this one was interesting because we decided to do a micro needle array. And the interesting thing, and you can see we built it, this is the actual device that was built and published. the big thing about microneedle arrays, I don't know if you guys know about BioLink, who's out there, and just got a Series A round. Microneedle arrays are really hard to get repeatable. And the reason is because it's really hard to get on the skin because all the different needles are going to go into the skin in different ways. And so you need to make these microneedle arrays repeatable and reliable. And so that was our first challenge that we tackled, and we tackled pretty well. And then we decided to do sensing. With this, we had to sensor, we had to extract it, and then sense it and detect it. And to sense it, we decided to use boronic acid and do a fluorescence-based sensor. And to be clever on that, we did ratiometric sensing. And then we built a really cool, very dual junction photodiode. So I know this may be out of the expertise of a lot of people here, but we were able to build something that could measure 2 different wavelengths so that, we don't have to worry about photo bleaching or anything, all on a little chip that's right here. So I'm going to go through that pretty quickly, and I want to get to the pumping work. So this is the hollow microneedle that we made. Again, people have made hollow microneedles for a long time, but the width of a human hair, you can see the array here. And then we have, the way we made it was we had little big squares and then tiny little pillars. So we called this a pillar, sorry, and these little guys fillers. And what was clever about what we did is you can see the top left here. Silicon is gray, and green is the silicon nitride mask, and blue is silicon oxide. And we made the pillar, you see this pillar at part C, I don't know if you can see my arrow, and D, we had the fillers up top. And what we did was when we, and these are the hollow, these things here are what makes the needle hollow. And what we did was we etched down the pillars and the fillers, and the fillers were so small. So what happens when we etched it down, then it would start attacking the sidewall. Once it attacks the sidewall, it could attack the side of the needle. So there was a play between diffusion and reaction. And again, a lot of people just make the needles following a recipe. What we did was undo everything and really look at, if you look at the right-hand side, the science behind it, and solve the chemical reactions behind it, so that we could make design guidelines for people to make very repeatable and reliable microneedles. And you can see what happened here. You can see in the bottom, filler, pillar, but based on the fillers, the pillar gap, length, height, ratio, all that stuff, we were able to really hone that in, and we hope that this will be really, really useful for people doing microneedle arrays, forever, for anything, not just glucose sensing. So after we built that, we started working on the sensing. And what you see here, the red star is the boronic acid. I didn't want to draw out all the chemicals. Boronic acid, when glucose comes in, it attaches to the glucose and it falls to this bottom thing, conjugated polymer electrolyte. You see the difference in red and blue here? So I'm going to play that again for you. We have a conjugated polymer electrolyte on the bottom, which is some fluorescence. See this blue? So it's some fluorescence we can see at a certain light. And then on the red is the right-hand side. And when the red hits the glucose and then comes close to the conjugated polymer electrolyte, it changes the emission. So it's ratiometric sensing. And what's cool about this format that no one's really ever seen before is that we can tune the band gap. We have 2 emissions, right? It's not just we're looking for 1 fluorescent signal, we're looking for 2 that are ratiometrically involved with each other. So we don't have to worry about photo bleaching, which is a big deal. And so it's in itself better signals. And we can do amplification. For those of you who know what conjugated polymer electrolytes are, there's something that allow for amplification. They've gotten very famous because of that. And we got a paper on this design, looking at this design. And it's this slide, you see the actual results. And you can see with 0 glucose, it's a lot of black on the right and the left and little on the right. And then as we get to 20 millimolar, and this is, physiologic concentration, 0 to 30 millimolar of glucose. So it was working. Great. And then this is something we just recently patented. My postdoc Bing Wang, who's amazing, went a step further. And actually, and this is what everybody wants. When you want to do glucose sensing, you want to have as many calibrations as you can. And he found this molecule, ARS, that actually, when bound, you can get an electrochemical signal. So when bound or unbound, you can either get a fluorescent signal or an electrochemical signal. So it's fluorescent, you shine light on it, depending on if ARS bound or not, you'll get a fluorescent signal. And depending on if ARS are not bound or not, you get an electrochemical signal. So you can measure both and self-calibrate based on that, because they should both give you the right answer with algorithms. And you can see on the right, we were able to do both electrical measurement and optical measurement with the scheme, and we just submitted it for a patent because we thought this was incredibly innovative for people wanting to go this route. And now, so now we have the sensor, it's about making the photodetector, and we literally just published this paper as well, where we have the silicon microneedle array, we put in an LED, and then we built from scratch our photodetector, silicon photodetector. And I didn't mention this, sorry, the sensing was done in collaboration with a incredible chemist, Guy Bazan, who is at UCSB and now is in Singapore. And this was done in collaboration with Dan Cohen in our electrical engineering department, who really helped, and he's now retired too. So we got really lucky with the two of them. And you see, we built this dual junction photodiode. And what you're seeing here is how we built it. There's a bottom diode and a top diode that have different wavelengths of light. And what you see here is the different powers. And the big thing is that it is sort of dependent on power, the wavelength of light and the current that comes out, but then we can figure out what wavelengths to use for the bottom and top. And what you see here is tartaric acid. We didn't unfortunately get it to work with glucose yet, but tartaric acid, we got it to work with 0 to 4 micromolars. And what you see is the black is the bottom junction, the red is the top junction. That's the photo voltage we're measuring. And the ratio changes. Look at just from 0 to 3. Is a different ratio as you get to different amounts of micromolar. Now, this needs a lot of work to optimize, but we showed all 3 parts, and I think every part of this has been very useful to companies and whatnot who want to use it with their work. But putting it all together would have been required of another 5 or 10 years of work, and we only had, this was after year 3, it was the end of last year, and we only had a couple more years of funding. And after all this work and realizing how much work we'd have to get this to fruition, and the fact that I had started a company called Lakshmi Therapeutic Devices that does not use microneedle arrays or neither, and it uses enzymatic sensing. And this company is actually already at a prototype stage. We figured that, and then going in humans, we figured that this company won, and I want to shift. This is the point of the Visionary Award, is just trying to make innovations, right and left to help people with diabetes. So I'm blessed that the ADA was willing to let me, work further to do, to now work on the pump, the integrated pump, and maybe even integrate it with the CGM. So I want to shift the rest of my time. I think I have another 6 or 7 minutes to tell you about what I've been doing this last year, which is all about pumping now. And again, I feel really blessed because we have a CGM that is smaller than a penny, that's going on the market, that's getting human data. And so I can start designing a pump with that in mind. And what's also amazing about doing pumping is it's right up my alley. I told you 13 years I spent doing fluid flow and micro and nano devices. And every pump out there, let's say the Omnipod, most of its space on the pod is the mechanics. We want most of the space of a pump to be the insulin. We want the pump to be the small part. So I want something that looks like this, where most of it's the reservoir and dispenser, and only a little bit of it is the actual physics, the pump. And I think we figured out a way to do that. And what you see here is through bipolar electrodes and nanofluidics. And this is in fact, the right-hand side is a chip I made in my postdoc years. I made this glass chip that has microfluidic channels with a electrode that's in the middle of the channel. They see the bipolar electrode. And what you see is a side version here, and the bipolar electrodes here. What we found in the last year is we did a lot of math and figured out the conditions to get pressure right in the middle over that electrode. And then we put a flexible membrane on top, and we could push fluid. And you see that here as a pump. If we have a bunch of bipolars, we can actually use this as a pump, and it's tiny. It's smaller than the penny, and it gives us the flow rates we want, which I'll show you. What you see in E is something that, CanMed is working on this, a new startup company doing bubble-based pumping. But we now can do it without any bubble generation, which is much safer, obviously, and much more repeatable. So to get into that, I want to teach a little bit of science. So what we're looking at here is a channel that, a microfluidic channel that is a glass channel. So it's negatively charged, which means positive charges are attracted towards the wall. You've rubbed glass against your hair, And you've seen positive charges wanting to go against that wall. I mean, you're negatively charged from the water, cations from the water, H3O plus ions. We usually use salt water in these channels. And then we have a floating. Floating means it's not attached to anything. We use Ti platinum. We use a ton of different kind of metals. When it's just hanging out here and there's no voltage, it's just, I just built this chip. It's mostly unpolarized. You see the electric doublers, that's the red stuff. Oh, sorry. And electric doublers do exist, right? But nothing's happening. When I put the voltage on, ding, I put it on, what happens? Well, the metal sort of shorts out, right? Electrons want to move towards the positive, the negative electrons in the metal wants to go towards the left, positive electrons in the metal want to go for the right. And then you have positive ions that are shielding the left and negative ions that are shielding the right. And so there's a cathode where electrons accumulate in the metal, anode where they are depleted, and it polarizes. So this is my voltage on, you see it polarized. And you have oppositely charged electric doublers that are forming at these poles. I hope I didn't lose any of you. I know it's hard to listen to the Zoom call like this, but this is what we showed. And this, one other thing is we tried to prove this with fluorescein. The problem is fluorescein quenches with pH. And if we're creating some electrochemical reactions, it's going to quench the fluorescein. So I just wanted to show this little figure here that shows that the pH goes down. Really, this is the BPE, and you can see that you'll get electrochemical reactions. So there's going to be a lower pH on the right. And that's why when you do the experiment, it's supposed to be really bright on the right and really not bright on the left because fluorescein is negatively charged. But you see it gets super dark here. We can actually completely recreate that with simulation. It's dark because it's quenching. You don't see this little, the really bright part because it's obscured by quenching, but it matches exactly our experiment, which is very exciting. And then we ended up doing it with other fluorescent molecules as well, but it wasn't as convincing, or this you can see the best. So I wanted to show that to you. So we published this because we understood the BPE. Now, what does that have to do with pumping? And this was sort of before we even thought about this as a scheme for pumping. Well, when my postdoc Alex Eden was doing this work, he realized that when you have the positive ions on one side, negative ions on the other side, they push together and cause pressure gradients. And so what do you see here? You see a very high pressure gradient, red is, high axial velocity, but you see, a lower pressure here and higher pressure here. So it's going to push up in the middle. And you can see the velocity is higher here, the velocity is lower here. And so what that means is you can actually, if with valving, you could actually make a flexible membrane on top of that BPE go up and down, up and down. And so you can see fluid pumped in with valves, or if the fastest valves are closed, fluid won't pump. And so you could apply a voltage back and forth and actually get pumping. And just to show that we did that, we did a lot of simulations where you see that, hey, if you don't have valves, you're just going to go up and down, up and down. But if you do have valves, you can actually get pumping. And now look at these flow rates. It's 0.6 picoliters per quarter of a second. So that's nanoliters a second, which is so like orders of magnitude more accurate, or than microliters that are in current pumps. And if we parallelize them, which is free, we could actually do a patch pump the size of a penny. And we did a lot of optimization here with amount deflection and flow rate and all this stuff on the right. I'm worried that I'm running a little bit low on time. So I want to jump to optimization, right? That was just showing a sine wave in the beginning. We can do the sine wave here, we can do an off square wave, and we can get different pressure diffusions and really optimize. And so that we're working on the experimental validation of this. Here is how we experimentally validate it. We have one channel, a membrane in a reservoir. You can see the actual device here. There's a PDMS slab, one channel with a PDMS membrane on top. And I'll show you some data now. What I think is clever is the biggest thing that I was asking my, this is a sort of side great result, where I was asking my grad student, okay, well, how do we measure deflection? I want to know how much this membrane actually goes up and down so we can match it with all the numerical simulations. And he came up with a really clever method. He came up with putting particles on the membrane and watching the particle go in and out of focus, right? And so what you see here is if it's 0, you see the particle, and as it's going in and out of focus, it changes. It's a point spread function. And he did this by calibrating it, and instead of just using regular particle tracking, he put machine learning in there, neural networks, to really learn everything here. And we got some very, very accurate data with deflection. And so what you see here is this is the BPE is here. This is particles in the channel. and you see the channel wall, you see a tracked particle, the distance from the wall, and we can look at how much things have gone up and down based on distance to the wall, and we can look at full deflection. And that's when the electric field is turned on, and we analyze it with geometric particle data. What you're seeing here, I'm just going to slowly finish up, is the peak deflection versus electric field. It's not exactly 1-to-1. It's a little bit more, the exponents more than 1-to-2 because of the science here, and don't have time to go over it, but you can ask a question and we can talk about it. I want to finish up. Lastly here is that we have, oh my God, sorry about my slides. The theory versus experiment works pretty well if we have a fit slope. So we are capturing it with our data. We're in the middle of writing this up and getting the last data to write up the theory versus experiment here. And lastly, this is the data from last week. I just showed the actuation of the membrane. We want pumping. And to do pumping, we need to have a channel above the membrane and show that fluid, insulin specifically, can move in that. And so what you see here is the first experiment that was done over the weekend, showing initial results of a PDMS lab on top of the PDMS lab. And you're seeing PIV here, and you actually see particles moving. So we get the fluid to move. I don't know how much pumping yet, but we actually show that this is working. So this is really exciting for us. I think it's a completely new pumping mechanism. We're talking with some people who, Steve Niani is the guy who worked on the Omnipod pump, who's interested in doing a pump, and a couple other pumping companies who are interested in this mechanism. So really exciting to get this out. So to end, I'm doing this for my daughter, and the Visionary Award allowed me to really use everything that I know to help her. And for the CGM, I could make repeatable microneedle arrays, which are going to be, I think, really useful. Ratiometric detection schemes, dual detection schemes, buried dual junction photodiode. We were able to make a BP patch pump actuation method, and a new method, or a better method, I should say, to detect small deflections. We want to, for the last year of the award, actually get trumpet curves for these pumps so that we can show this is a valid pumping scheme and people can use it for insulin delivery, and initial data so we can spin something off and use everything we've done with the Visionary Award to make a difference. Okay, so I think I stayed close to 20 minutes. With that,
Yeah.
I thank you all, and I can take questions.
Thank you, Sumita. That was really very interesting. We have questions from David Nathan and Gokhan Hodamichlagil. I'm going to, David and Gokhan asked a question in common, so let me start with that. Have you gone through testing in animals? And how far down could this be scaled for small animal work?
Okay, so that's great. I'm seeing it. So CGMs are generally not tested in animals unless we think that the materials are going to be toxic. But most CGMs are tested directly on humans. So what I can tell you is that the needles that are in Lakshmi have been tested on me already, and we're getting IRB approval for going straight to humans because every material we use have already been FDA approved. In terms of the pump, we're working on cannula materials that we can coat to get rid of the phenol and other abstracts. That's another thing I didn't get to talk about. And that we will be testing in pigs first to make sure that that works. And then the second question about concentrated insulins. Yeah, we've talked to Lily. We are working right now. We think that we also have thermalin insulins. So I think Karen Tomlash asked a question about insulin in these materials. So I had a JDRF grant a couple of years ago that we looked at the interference or the interaction between the insulin and the material, how much insulin aggregates. I can tell you PDMS is terrible. It's very hydrophobic. Insulin aggregates everywhere. You want to try to avoid that material, but so that this is just a prototype and we're looking at coatings for it. We've done a lot of work and I can show you that data. In fact, I think it's published somewhere for everybody to be out there, where we've used thermalin U1000s and U400s and U200s from Sanofi and looked at that interaction. Of course, they aggregate much, much quicker. This pump, I think we can survive with U100 for a few days, and then as new insulins get on the market, we will transition. I'm worried about how long it's going to take for the insulins to get on the market. As I said, I want this all to be done before my daughter goes to college, so I only have 8 to 10 years, and I'm worried that if I want to do something with real insulins that are concentrated, it's going to be more like 10 to 20 years. So that's what I'm worried about there.
So let's one last question, I think, and we'll have to move on. So David asked about specificity, and that links to a question that I had. the specificity of these sensing devices for glucose. For example, does boronic acid interact with other sugars besides glucose,
Definitely does.
or any related molecules, and what kind of problems does that cause for this technology?
It definitely does. It definitely does. And this is one of the reasons why we moved on to the pump, because we think that the schemes of the ratiometric sensing and the dual sensing are really useful. But what I'm doing at the company with enzymatic sensing is so much more accurate and sensitive that this doesn't seem like the viable path forward. Exactly what David said. However, I think it's really important to publish and show, because ratiometric sensing and for lactate, for all the other ascorbic acid, everything else that we want to test in diabetes, this could be a useful scheme. But certainly what you're saying, completely agree with, which is why Lakshmi started using enzymatic sensing.
All right, well, we're going to have to end there. Sumita, thank you so much for a very stimulating and exciting presentation. And Marie-France, are you in the process of showing your screen?
Yes, I can. I'm just going to start.
Wonderful. Okay, so while she's getting that up, let me introduce Marie-France Hivert from Harvard. And her presentation is about gestational diabetes and epigenetics. Take it away. Thank you.
Can everyone see my screen correctly?
Yes.
Thank you. So I would like to thank the ADA for inviting me to talk in this Pathway Award series. I am one of the 2015 awardee, and I had the chance to have an Accelerator Award, and I'm really, really grateful. That's definitely was a huge part of my early career. So today I will share with you some of the most recent emerging evidence on the links between epigenetics and gestational diabetes. But as you will see, the associations may not be in only one direction. I have no conflict of interest to disclose. Many studies are suggesting that there is a transgenerational vicious cycle where adverse metabolic in utero environment lead to altered fetal growth and metabolism, somehow programming the child for excess adiposity and all its metabolic consequences, mainly type 2 diabetes. In my lab, we investigate how maternal hyperglycemia can lead to this programming. And one of the questions we're trying to answer is, what are the determinants explaining this transmission of obesity and diabetes? One of the obvious hypotheses is it might be genetics. If a mom is carrying genes that increase the risk of obesity and diabetes, then her child is also carrying part of this genetic burden. Others would say, well, it might be because of shared environment and family choices, such as diet and activity, that have all been linked to diabetes. But there are studies that suggest that it might be more than genetic or familial environment. Here in this study of OHEM population, the Bill and colleague more than 20 years ago, using a sibling pair design, one sib was exposed to maternal hyperglycemia because the mother had developed diabetes before pregnancy, and the other sib was born before the mother had developed type 2 diabetes, and so was hypothetically not exposed to hyperglycemia, at least to not the same degree, during in utero development. So this design is very clever because it does control for familial risk and partly for genetics. And you can see here on this graph that sibling exposed to diabetes during in utero development in the black bars had much higher BMI, especially during the teenage years. They also found that siblings exposed to diabetes in utero had a much higher risk of diabetes themselves, with an odds ratio of 3.7. Based on this study and many other epidemiological observations, more and more investigators believe that exposure to hyperglycemia during pregnancy leads to this programming of future metabolic disease, and became interested in revealing the molecular mechanism behind this metabolic programming phenomenon. Such biological mechanism needs to be malleable by environmental exposures, such as the maternal glycemia here, but also durable and have a long-lasting effect, leading to chronic diseases over the life course. And more, we believe that epigenetics could be one of the molecular mechanism involved in metabolic programming. Epigenetics is the study of variation in gene expression caused by a mechanism other than the underlying DNA sequence. And specifically, most epigenetic phenomenon are methodically stable and enduring and can influence expression over a long period of time. And some human studies have even demonstrated that specific epigenetic marks may be stable over decades at specific loci, while other sites seems to be more malleable. Indeed, epigenetic phenomenon can be modulated by environmental factors such as pollutants, diet, smoking, either pre or postnatally. But there's evidence suggesting that there is a particularly sensitive period in utero, and this makes sense in the context of rapid cellular replication and tissue differentiation during fetal development. Classically, the two main phenomenon that are considered epigenetic mechanism are histone modification and DNA methylation. Some include also microRNA as epigenetic regulators. Today, I'll focus mostly on DNA methylation since it's the most commonly studied phenomenon in the context of fetal programming. With the recent and rapid advances in technology, we now have arrays that allow us to investigate a large number of methylation sites across the human genome. And the most recent array is called the EPIC array from Illumina, and it covers over 850,000 CpG sites. So given the possibility to interrogate thousands of CpG sites using a simple array, many pre-birth cohorts have tried to investigate how gestational diabetes is associated with DNA methylation in the offspring, most often using cord blood samples given the easier access. Unfortunately, many of the early epigenetic association studies included small sample size with less than 30 cases of GDM, and almost no hits replicated across each other in the early reports. But also, fortunately, similarly to the genetic field, many investigators in the field of epigenetic and developmental origins of health and disease have now joined forces in consortia. And here is a recent report from the PACE consortium, where we meta-analyzed data from 7 pregnancy cohorts, which included almost 4,000 mother-child pairs, with 317 cases of gestational diabetes. Those numbers are still very small compared to any genetic consortium, but much larger than prior epigenetic reports. For this meta-analysis, we use cord blood DNA methylation and the prior Illumina array, which covered around 400,000 CpG sites. You can see the result of the meta-analysis here using the classic Manhattan plot with the 22 autosomal chromosome on the x-axis and the strength of association using the negative log p-value on the y-axis. The red line up here indicate our threshold for statistical significance using a false discovery rate of 5%. And despite being the largest study so far, we actually did not even surpass that 5% FDR threshold for statistical significance. So we went on and conducted regional analysis to try to gain a little bit power in using statistical approach validated in the field. We identified 2 regions that were consistent across cohort. One of them is OR2L13 and the other one is CYP2E1. And what are those 2 genes? The first one encodes for an olfactory receptor, and this is the only one CpG that we replicated from prior case control studies. One CpG in that region has been reported before in a small case control GDM association study in cord blood. Interestingly, the same CpG site has been identified as differentially methylated in blood and buccal cells of a case control study of autism spectrum disorders. And there's been some epidemiologic studies showing association between maternal hyperglycemia in utero and autism. So maybe future studies could investigate potential mediating role of methylation at that loci. This CYP2E1 encodes for a member of the cytochrome P450 superfamily, and these enzymes catalyze many reactions, including synthesis of cholesterol, steroids, and have a key role in gluconeogenesis. This protein is also highly expressed by the liver, can be induced by hyperglycemia, starvation, and has been associated with various liver conditions, including fatty liver disease. One thing we have to keep in mind is we were unable to replicate any other of the CpG sites previously reported by small case control studies that were published in recent years, even with less stringent p-value threshold, which supports the importance of replication studies or meta-analysis of multiple cohorts for these type of analysis. And despite many strengths to our study, we also had many limitations. GDM was diagnosed using different strategies and criteria, and GDM treatment was likely implemented in different ways, depending on the country and the years of the study. So in theory, if it is the maternal glucose that programs the fetus, the treatment of GDM could lower the hyperglycemia and so dampen the effect of programming, but it's hard to evaluate as none of these cohorts had detailed measure of how tight the glycemic control was in late pregnancy. So to address the limitation of gestational diabetes as a categorical exposure and the different criteria, as well as the intensity of treatment after diagnosis, we can investigate maternal hyperglycemia as a continuous exposure variable. And as a reminder, the HAPO study very well demonstrated that maternal hyperglycemia is associated with risk of complication in the linear fashion with no lower threshold. So in my lab, we investigate the association between maternal hyperglycemia and metabolic programming, leveraging physiology and glycemic measures along the whole spectrum of glycemia. To do so, we have built a prospective pre-birth cohort study called Gen3G. And between the years of 2010 to 2013, we recruited over 1,000 women in their first trimester, and their characteristics were similar to women receiving prenatal care at our institution. We followed them up at 24 to 28 weeks of gestation, where they all did a 75-gram oral glucose tolerance test. We found that 8.4% of women were diagnosed with gestational diabetes, which is an incidence expected given the population we had recruited. And we also measure insulin at multiple time points of the OGTT to calculate indices of insulin sensitivity and insulin secretion. We then followed women at delivery, where we collected clinical outcomes. The characteristics were once again as expected for the population we had recruited, and we collected placenta and cord blood in over 700 deliveries, allowing us to do a lot of the work I will be presenting in the next few slides. So focusing now our attention on the placenta, it is well known that maternal glucose crosses freely the placenta and then stimulate insulin for fetal growth. But the placental adaptation to maternal hyperglycemia are still obscure. And so we conducted an epigenome-wide association study of maternal 2-hour glucose in DNA methylation of the placental tissue using the most recent EPIC array to investigate over 800,000 CpG sites. This is a summary of our main finding, again, using the classic Manhattan plot. The red line this time indicates a Bunfer-Erny correction, so this is a little more stringent. And then the yellow dotted line, the 5% falls discovery rate. In this analysis of maternal 2-hour glucose with methylation in fetal placenta, we identify 4 loci that reach the 5% FDR, but our most important loci was at PD4B, where we had multiple probe that were identified even above the Bunfer-Erny threshold. Other loci of interest is the TNF receptor SF1B, and another probe was identified near the LDL receptor. This fourth here, BLM, is a gene with little known function. We then pursue our investigation looking at a little bit more of functional study. We look at expression in each of the locus, and we were happy to see that all 4 probes at PD4B, their methylation level were associated with expression of PD4B in the placenta. We also found correlation between methylation at the main probe of interest at the TNF receptor and the LDL receptor with their respective genes in the placental tissue, but this was not the case for the 4th loci. But what is PD4B? PD4B is a phosphodiesterase responsible for the hydrolysis of cyclic AMP. And we know that PD4B decreases TNF alpha secretion, which is very important in the context of placental biology, and is a putative role in inducing physiologic insulin resistance during pregnancy. From animal models, we know that PD4B inhibition in pregnant mice has been shown to block intrauterine inflammation, decrease cytokine production, and delay preterm birth. PD4B null mice have been shown to be leaner with lower fat pads, smaller adipocytes, and lower leptin levels. And chronic treatment of PD4B inhibitors was shown to delay progression of diabetes in DBDB mice. In human studies are more scarce, but RCT of patients with recent type 2 diabetes demonstrated that treatment with PD4B inhibitor successfully lowered glucose level. So these evidence are really supporting that we might have identified an important loci that may be implicated in programming of long-term adiposity and glycemia. Now moving forward to another analysis, we focus our attention on an important part of the maternal physiology, which is insulin sensitivity. And this time we move forward thinking about the 2 direction of the potential association. And you'll see that it's not only the maternal physiology can influence the DNA methylation in the placenta, but maybe also that the placenta epigenome may have a potential contribution on gestational diabetes pathophysiology. So as I said, the placenta is well known for its nutrient transfer role, and but we also know that it's a very active endocrine organ and it can have important influence on both fetal and maternal physiology. And one of the major physiologic change that occurs during pregnancy is the dramatic decreases in insulin sensitivity, especially in the second part of pregnancy. Yet we know very little about the placental factor responsible for this reduction in maternal insulin sensitivity, since the most commonly placental hormones correlate poorly with insulin sensitivity. The placenta also adapt to this change in maternal insulin sensitivity and thus impact the fetal development. And this tug of war between maternal and fetal energy requirement is often described as the maternal-fetal conflict. So in theory, this decrease in insulin sensitivity is to allow more nutrients to be derived towards fetus, but there's also some reaction from the placenta. And to investigate further this placental adaptation or role in insulin sensitivity during pregnancy, we again investigated the whole epigenome in our Gen 3G cohort. Once again, I'm showing the results of our epigenome-wide association study, this time looking at the maternal insulin sensitivity index and placenta DNA methylation. And again, the red line is the Bun-Ferroni threshold. And you can see that we found a lot more signal. This time we actually identify 188 CpG sites that reach Bun-Ferroni. Some of those loci are worth of mention. One of them is TCF7L2, which is the most known genetic loci for risk of type 2 diabetes, and also genetic variants at this locus has been identified as a genetic risk variant for gestational diabetes. Another probe was identified near the prolactin receptor, highly relevant during pregnancy. Another locus example is PRDM16, which is a DNA transcription regulator key to differentiation of brown adipose tissue. And then another example, it was a probe near KCNQ1, which is not only a well-known type 2 diabetes risk genetic variant locus, but was shown to increase risk of type 2 diabetes in a parent of origin transmission fashion, in line with the fact that this locus is a known maternal imprinted gene. KCNQ1 is not the only imprinted gene that we identify in our top hits. In fact, we identify 12 known placental imprinted gene, 9 that are known maternally imprinted, meaning the paternal allele is the one expressed, and 3 that are known paternally imprinted, meaning maternally expressed, including methylation at H19, which is located nearby IGF-2. That is, IGF-2 is the other main growth factor during in utero development, in addition to insulin. IGF-2 is paternally expressed, against fueling this maternal fetal conflict hypothesis, where the paternal express IGF-2 should be promoting fetal growth, and maternal express IH19 in the same region would presumably limit fetal growth to preserve maternal fuels. This locus also include microRNA 675, and its putative role is to limit placental growth via reduction of the main IGF receptor. To try to untangle the direction of effect between the placental DNA methylation at all of the CpG sites we identify and maternal insulin sensitivity, we applied a bidirectional Mendelian randomization to try to know, is it the maternal insulin sensitivity that influence the DNA methylation on the placenta, or the placenta epigenome that tried to regulate maternal insulin sensitivity? To do this, we were able to build genetic risk score for 131 of the CpG sites methylation levels that we had initially identified. And based on those genetic instruments, we found that placental DNA methylation seemed to be causally implicated at 5 of the CpG sites identified, including one here, DLGAP2, which is one of the imprinted gene that I highlighted in the prior slide. DLGAP2 is biallically expressed in the brain, but only the paternal allele is expressed in the testes and in the placenta. One more downstream analysis that derived from that analysis is that we noticed that among the list of genes annotated to the 188 CpG sites that we had identified in our primary analysis, we were intrigued to find many other microRNAs in addition to the microRNA 675 that I highlighted. And so we use pathway analysis in microRNA target scan and found strong enrichment for targets of multiple microRNAs. They suggest an important role for microRNAs in placental biology and glycemic regulation during pregnancy. Overall, our findings support placental DNA methylation signature is fundamentally linked to maternal insulin sensitivity. We identify CpG sites at which our Mendelian randomization supports that placental DNA methylation have causal influence on maternal insulin sensitivity. And the identification of both maternally and paternally imprinted genes is in line with the maternal fetal conflict hypothesis, suggesting that genes from both parents try to regulate maternal insulin sensitivity during pregnancy. And finally, the enrichment in microRNA targets adds the recent literature in implicating that microRNAs might be having paracrine or endocrine action. To conclude, I hope I was able to demonstrate that epigenetic may be part of the programming of the offspring metabolic future, but also that the placental epigenome may participate to regulation of maternal insulin sensitivity, thus contributing to gestational diabetes pathophysiology. In the future, we are continuing to push further our investigation of placental genomic regulation with our current NIH funding to study placental RNA and microRNA and try to better understand the placental role in maternal insulin sensitivity, insulin secretion, and risk of gestational diabetes. I want to acknowledge my research team and collaborators in Sherbrooke who have been instrumental in building and maintaining Gen3G, all my colleagues at Harvard and outside, including the multiple consortia. And of course, I want to acknowledge all the funding agencies, especially the ADA Pathway to Stop Diabetes program that were definitely central to all our works in genetic and epigenetic studies.
Thank you very much, Marie-France. That was really elegant. And we have a couple of questions. I'm going to take a subset for you. First from Steve Parker, one of our awardees. Very nice work, Marie-France. Are your locus gene names like in GWAS, where it's really a marker for the region, or do you think that that's the effector gene at the locus? I'll just stop there. I'm sure you understand the gist of the question.
Yeah, so absolutely, just like GWAS, especially using array, a lot of our findings, the annotation is just linked to the nearest gene. And that's why we conducted the expression study in our placenta. We have the chance to have a tissue that's relevant to what we're studying. And so by doing the methylation to expression in the same region, and we did a few genes around that, the main sites of interest, and the one I showed you were the one that were associated strongly with the methylation at those sites. So we know the limitation of GWAS and EWAS. We try to do a little better to get at the functional part, but you're absolutely right that it could be other genes around that CPG sites.
Okay, I'm going to move forward. Chuck Burant asks, are there any studies that ask whether genetic variations change the susceptibility to methylation in promoters? For instance, are TCF7L2 susceptibility loci related to the degree of methylation?
So we looked whether the known genetic variants at TCF7L2 were associated with the level of methylation in our placenta, and they were not. And so in this case, and we would need to go loci by loci, but in this case, it's and they were not that close in genomic location, the actual variants and the methylation, but it is known that in many instances, especially in promoter region, there is genetic variants that are strongly associated with DNA methylation.
Okay, great. And a question from Steve Rich. Are there data indicating other medications that the mother is taking could affect DNA methylation, chromatin accessibility? For example, role of stress and anxiety or anxiety medications on methylation?
So we did, not in Gen3G, but in another cohort, we looked at some medication, SSRI, and we found a couple sites in the cord blood that were differentially methylated, but they were definitely not in the same region that we found here.
Okay, and just as a general knowledge question, how does placental DNA methylation relate to methylation in other tissues in the mother? Because, and the reason I asked that question is I'm fascinated by your PDE4B
Mhm.
result, because, for example, PDE4B could have a regulatory role in insulin secretion from the islets via its ability to control cyclic AMP levels.
So the placenta is a very unique organ, and during in utero development, so at the very beginning at fecundation, there's erase all the DNA methylation marks except for the imprinted gene, and then there's a remethylation, and the placenta remethylate, but not at the same level than the other tissue. There is still some correlation at some loci between some tissue, and we've been able to look at the correlation between cord blood and placenta, but there is definitely not like a data set that I can look for like placenta and islets or liver, because you
Right,
can't
right.
really biopsy those in newborns.
Okay,
But
thank you. And a last question from Richard Bergman. Is there good evidence that OGTT insulin sensitivity is accurate in pregnancy?
so we selected the Matsuda index based on our RGTT because Pat Catalano in the past has validated that in pregnant women against clamps. Of course, those were small numbers, but among all the indices that he tested, the Matsuda was the most reliable compared to the clamp data.
Okay, great. All right. Well, in the interest of everyone's busy schedules, I think I'm going to call it to a conclusion. I want to thank our Marie-France and Sumita for beautiful first lectures, really diverse topics. And I think that's going to continue as we go through this series. There'll be a lot of diversity in topics. And I hope that we get as good a turnout as we got today. We had about 50 people on, and it's just, that's essential for the success of the series. So please plan on coming back. The first Wednesday of the month, and our next lecture will be January 6th of 2021. Jonathan Sweedler and Paul Cohen will be giving the talk then at 2 p.m. on January 6th. And I strongly encourage everybody to keep coming. I think this can be very, very valuable. So with that, I'll say thank you. And if you don't have a complete schedule for the 2021 series, we will be getting that from Kylie Goldstein here shortly. Take care, everyone, and thanks for joining. Bye-bye.